I'm going to talk about an aphid salivary protein that controls plant autophagy. So as you all know, aphids are sap-sucking insects that which are responsible for major crop damage all over the world by both consuming plant nutrition and by transmitting viral diseases. So similar to other phytopathogens, insects also manipulate plant physiology by delivering a cohort of proteins and small biomolecules such as hormones into the plant cell, and through which they control physiology and manipulate and compromise plants' defense. If you put a bunch of aphids on a bunch of aphids on tobacco plants and look at them after 2 weeks, a couple of weeks, you can see that the plants look like, compared to the ones that didn't have aphid, the plants have basically look like they have stunted growth, and they basically look like zombie plants that are just factories for food for aphids. And the way that aphid feeds on the plants is by through its stylet, when it actually pokes through the plant leaf and basically poke through in and out of each cells until it reaches the phloem sap, where it starts feeding. And through this poking and probing, it basically secretes salivary gland proteins into the plant cell. So this mechanism, especially in case of aphid, the nature of these molecules and their function is very poorly understood. So I took advantage from these characteristics of aphid, that when it feeds, it mixes the saliva with the diet. And I reared aphids in these little cups covered with 2 layers of parafilm. And then I put some artificial diet, mostly sucrose and water, between the 2 layers, and allowed them to grow for to feed on there for 2 days. And after 2 days, I collect them and did metabolome. It's hard to go between these two. So did the metabolome and proteome with collaboration with my husband, Naveed. And we basically took these proteins, found the proteins, and I selected a subset, a small subset, like 48 proteins, which had signal peptide, which is characteristics of proteins that are secreted out of the cell, and is the characteristics of most of the effector proteins, basically. And I cloned them and expressed them, cut the signal peptide off, fused them to GFP, and expressed them in bentamiana leaves. And then first thing I looked was the symptoms, what they do on the cells, on plant leaves, and most of them were like similar to the GFP control, no symptom. Some of them had some slight discoloration, and 3 of them, interestingly, were showing this white patchy type cell death response. So I started to look at those 3 specifically. Also, I want to mention that they had, because they had GFP, I could look at their subcellular localization, and they had very interesting localization. For instance, this protein was targeted to plant cell nuclei, and another protein was targeted to ER, and it was causing this overexpression of the protein was causing the cell death. And the 3rd protein, which is a basically topic of this talk, SP11, I call it, was causing cell death, but also was located in these punctate little structures, which were co-localized with the autophagy marker ATG8. So, what is autophagy? Autophagy is described as one of the 3 types of cell death. However, in autophagy community, it is not really considered a cell death, similar to necrosis or apoptosis, because it doesn't really kill the cell. It's just a process that's ubiquitous in eukaryotic cells, all eukaryotic cells, and it's degrading the cytosolic content. Depends on in response to different physiological and developmental needs of that cell. So what it does is that it's the hallmark of autophagy is first is this the bilayer membrane, that the 2-membrane organelle like vesicle, which sequester plant contents, the cell contents, including organelle, the cytosolic content, could be mitochondria, nuclei, chloroplast. It starts with this isolation membrane, and it's speculated that this isolation membrane is initiating at the ER, endoplasmic reticulum. And it's decorated with this particular protein family called ATG8, which is a protein that needs to be lipidated, and the lipidation is very complicated process, needing a lot of protein. proteins. I'm not getting into the details of them, but once it's decorating this vesicle, this large autophagosome vesicle, the vesicle moves and merges with vacuole plant and yeast or animals, in animals with lysosome. And eventually it degrades the material inside is degraded inside the vacuole and turned into building blocks. The person who discovered this pathway basically got Nobel Prize in 2016 because it's very important in medicine. As I mentioned, these little like circles that are moving inside the vacuole, coming and disappearing, were indicator of autophagosome or autophagy bodies, and they were co-located. When I co-express SP11 with GFP and mCherry with ATG8, which is that adaptor marker, they both were in the same place. So we knew that this protein is, we found the protein in aphid saliva. However, I wanted to see if it's actually secreted by aphids into the plant. So I raised an antibody against the protein, which could recognize the GFP fused to the protein on the infiltrated leaves, also could recognize the native protein in aphid's body, and I could recognize the protein inside the plant cell after aphid fed on the plant and not on the plants that didn't have aphid on them. This is a non-specific plant. So next I wanted to see if this protein is helping aphid or is helping plant to defend against aphid. To do that, to measure that, we do a bioassay, and one of the bioassay ways is to look at the reproduction of aphids on plants that had the protein or not have the control. And it turned out that unlike our expectation, against our expectation, the plants actually that had the protein, aphids grow much better on them and reproduce more. So I also knocked down the protein inside the aphid body using RNAi, using two different constructs. This is the gene expression, indicating that they were knocked down. And in collaboration with Hong Lin, a former member in our lab, we did some survivalship assays. And it turned out that the plants, the aphids that didn't have SP11 or had less SP11, their survivorship was decreased relative to the plants that didn't have the RNAi construct. So this so far indicated that SP11 is secreted by aphid into the plant cell and is helping aphid and is co-localized with autophagy marker. But in order to understand what it actually does, the mechanism of this protein function, I needed to look at the partners. First thing that people look is usually is like, what is this protein binding to? So I did some co-unit precipitation of SP11 fused to GFP and pulled down the protein from the plant and try to find the plant partners. Two groups of proteins came down with SP11. One group was these two proteins, RABD1 and RABD2A, which RABs are basically ROS-related small GTP binding proteins, which are proteins that are essential for vesicle trafficking all over the cell. This is a big family of the proteins. Interestingly, these members that I found actually were implicated in autophagy initiation and maturation of autophagy in yeast, in mammals, and this particular homolog was found in Arabidopsis to be essential for autophagy. Recently was published. Also, it was shown that in other oomycete, in case of another phytopathogen, there is an effector that binds to the adaptor here. It's basically a receptor for the adaptor of autophagy, ATG8, and also binds to the RAB, and it basically recruits the lipid droplets to the site of oomycete infection so that there is more starvation-induced autophagy in that spot. And the way that effector pulls lipid droplets is through via this RAB. So RAB was a great candidate. The other candidate was this phosphatidylinositol 3-phosphate transfer protein, which is basically a type of lipid, a specific lipid transfer protein. And it's unknown, its role in autophagy so far is unknown, but we know that from literature that this lipid, the area of the ER, which is speculated to be the origin of autophagosome formation, is enriched in this lipid. So these two were great candidates to begin with. SP11, as I mentioned, was co-localized with ATG8, but I also wanted to see if it's co-localized with RAB. And as you can see here in these little dots, which are the place where autophagosums start, that they were co-localized there. I also did some reciprocal co-immunoprecipitation, this time fusing GFP to RABs, the two RABs, and could pull down when I co-express with SP11HA or FLAG, I could pull down the two RABs and not with the free GFP. And when I overexpress RABs together, RABs alone in tobacco, the symptoms that was shown here in Bentamiana, the symptom was similar to SP11. Indicating that they most likely are in the same path. I also looked at those adapters to see which isoform of those adapters actually ATG8 may bind to SP11, and these adapters are membrane proteins, they are very hard, and one of them seemed to be having some interaction, not as good as RAB, but some interaction. These are the adapters as bait, and this is the SP11 that was pulled down with them. So I did some molecular cloning, and at the end of the SP11, there is a domain that is predicted to be RAB binding or activation domain. So I made a truncated version of that SP11 protein, and as you can see in the truncated versions, that cell-led response disappeared. And also, when I looked at the punctase structures in those fragmented RABs, the punctase started to disappear, and basically protein became cytosolic, or like free GFP, it went to the nuclei, indicating that this domain, RAB binding domain, was essential. And when I used RABs fused to GFP, each one of these pairs are RABs, as bait, I could only pull down SP11, full-length SP11, and not the truncated versions in the pull down, indicating that this domain was essential for the function. My reciprocal co-IP also proved that this protein, which was the phosphatidylinositol 3-phosphate, could pull down SP11 as RAB did and as that ATG8 isoform did. And because all of these are lipid-binding proteins, and previous literature showed that RAB may recruit lipid droplets, I stained lipid droplets using BodyPy, which is green here, and to see if actually I can see them bind to those red punctates, which are SP11 here, RFP, and seem to be like co-localized with them. And finally, the final question is, this effector so far, okay, we know it's binding to proteins that are in the autophagy pathway, but does it suppress or induces autophagy? And the most standard marker for looking at autophagy flux and induction is to look at ATG8, that adapter that I mentioned, fused to a tag, either GFP or M-cherry at its N-terminal and not C-terminal, because C-terminal is lipidated. And then look at the ratio between free GFP or M-cherry and the full-length protein. When the autophagy initiates and induces, basically, the ATGs go into the vacuole and they degrade. However, the tag, for some reason, these tags are more stable. And if you see the overaccumulation of tag versus the full-length protein, that means that the autophagy is induced as initiated. And when I did that for both cases of dark and light, I saw that this overaccumulation of the tag relative to the full-length protein, indicating that most likely autophagy is induced by this effector and not suppressed. So with this, our current model is that basically the effector is somehow binding to these lipid binding proteins, enriching this membrane, the initiation membrane, which is the big question in autophagy field. They know that it needs a lot of lipid remobilization, but the source of these lipids and the mechanism of this lipid remobilization is completely very poorly understood. So I think that AFID is basically inducing a starvation-induced autophagy and send these autophagosomes to vacuole to make the amino acid, lipids, and sugars, which are building blocks for its feeding. Basically, AFID is using leaves as a second gut. And with that, I would like to thank my collaborators, Navid, for a lot of mass spec, Honglin, Sergey, and Venkatesh for great suggestions and helps, and George Jander, my boss, who hosted me in his lab, and he has unlimited support. Thank you very much, and the lab.
Thank you, Leila. I think we have time for one or two questions.
Yes.
Great talk, Leila. This
Thank
is
you.
amazing. I was wondering, did you ever look at the RAP mutants or silencing RAP? Is that enhancing the aphid feeding? And then is it possible to kind of make plant resistant to aphids by complementing the RAP mutants by a RAP version that doesn't bind to your SP11?
Those are great things that I need to do. I mean, RABs, basically the mutants in Arabidopsis, at least one for the two RABs, I thought that somehow they have redundancy. I ordered one, but I couldn't find the other one. And well, there's some autophagy mutants too. That I tried doing aphid bioassay on them. But keep in mind that autophagy mutants basically are compromising all sorts of autophagy. And there is like this community is saying starvation autophagy versus defense autophagy. So it's not really easy to, I haven't got any real solid conclusion about that. However, I knocked down, I believe like a couple of years ago, both either RAB or autophagy in tobacco, and like that cell death was less, the response was less. So yeah, that's a part. We're trying to publish this because it's like we are in a hurry. And then, yeah, definitely that's like a lot of questions in future, but that's great. Thank you.
Are there any other questions for Leila? I guess I had a question.
Yes.
Are there any plants that are kind of like resistant to aphids? And is there any possibility that there's like a plant defense that kind of prevents kind of SP11 from getting into cells and like blocking that potentially?
Well, it's a
Sorry.
very hard one. Like you want SP11 not to get in the cell, so the aphids should not be able to deliver that.
Yeah, or
that's
is there
on
like
the aphid side. But in terms of like if there is plant that's very resilient, actually tobacco is very, very resilient. Heat. And like aphids hate tobacco, not tobacco, bentamiana, not tobacco, actually they like. But we have a mutant in the lab, which is acyl sugar mutants. And that's where I did those bioassays. So that if you knock out acyl sugars completely, then aphids like kill bentamiana. And so, yeah,
Okay.
those are Georgia's expertise, like metabolites.
Yeah. Awesome. Well, let's thank Leila. Trevor, is yours loaded on here too?
Yeah, probably. It's on the desktop. It better be because I don't have it on.
Let me ask IT potentially.
But also I think there's a break between upper right corner.
Wait,
and
I
desktop.
found it. Thank you.
Trevor was saying there may be a break now.
Yeah, what time does the next talk
I
start?
think we're doing, sorry, I gotta check my list. So I
Just so
think
people
that
can-
you're at, yeah, so it's you and then a break at 10:30.
Yeah, but at 10:15 is when. Starting.
Oh, shoot, okay, sorry, I'm going, yeah.
Session, we need to
Oh, okay,
coordinate.
perfect, great, okay, sorry, I thought it was 10:05.
It's all good.
Any other questions for Leila? We're done than I thought.
She's back.
I was so worried. I'm over time. Usually I am. Sorry.
No worries, no, there we go,
I got a
okay.
question. Is sort of the inducing of autophagy, is that a common strategy for insects preying on plants, or do you know?
Inducing autophagy, so far, based on the published literature, the only phytopathogen that I know of that has an effect or something to induce autophagy is that Phytophthora-omycetes. And it's like tons of papers are published from Sophie Cammons and Tolga Boskard in UK lab. And not in others, like autophagy is very complicated. So to find that whether it's actually really is inducing autophagy, it's kind of like hard. And also keep in mind that autophagy is like something that happens in all of our bodies, depends on our responses to food. If you are starving, the autophagy is induced. If plants are in dark, the autophagy is induced. If a pathogen attacks, plant defense includes autophagy. It's not just the pathogen, but also the defense. So it's like in case of Phytophthora, basically they showed over 10 years that the pathogen is diverting the defense, little autophagosomes, defense-derived little autophagosomes towards their side to turn them into a starvation-induced autophagosomes, which are bigger. So that basically it's replacing the receptor in those autophagosomes, not the adaptor, the receptor that binds to cargo. It replaces the plant's native receptor to its own receptor, to whatever that effector was replacing that, going to those autophagosomes and replacing them, basically changing their nature. So yeah, it's kind of like.
I have another random question.
Sure.
Leila, I might have missed it, but I was curious, what is this protein like SP11? Like how?
It's very unknown structure, like it's a small protein, but just like it has, you cannot find what it is, like, it's quite unknown, except for that last domain that I told you was RAB activating or binding domain. I don't know anything about other than. And aphids have 200, 300 proteins that are completely unknown, at least like based on genome, that doesn't exist in any other species. So I'm guessing there are lots of convergent evolution for particular pathways. It's one of the proteins that you may have in other species that does the same thing, but it's like has some domain or something, but it's not in aphid. It's completely only unique to aphid, and I think brown hopper probably has that too. Yes.
So OpenAI now has this collection of three-dimensional structures of supposedly all the proteins that exist. Have you looked at to see whether that's available?
I haven't, but I think I exhausted many of this modeling and prediction ones, but not that one. I can look. Yeah.
I was shocked if that was one of the things they said. We have over the last several months determined the three-dimensional structure of every protein that exists, supposedly.
Yeah, it's like an AlphaFold, I believe, right? That's like I don't know what they use, but I tried really to find out what. But it's amazing that there are some proteins that in, for instance, aphid or other insects that doesn't exist anywhere else. So one of the characteristics that I chose to put as an effector was that a protein that's not in all the bugs or all the insects. It's unique to like the sap-sucking ones, or it's unique to that kind of like category. Yeah.
I have another random question. So this SP11, if you do a tobacco transient leaf transformation, not only in tobacco, but in other plants, is it inducing the same symptoms to kind of like look at natural variation in responses to SP11 through like species approach?
So that's a good question. So I put the protein in Arabidopsis, for instance, permanent transformation. And it doesn't, like you can see some leaves have this whitey patchy, but actually the plants grow better. And when I was in a conference on like autophagy conference, which are like all autophagy people, which I couldn't even, I was so scared. They were like all saying, wow, you're saying it's cell death. Cannot be autophagy actually sometimes help grow. Like they are confused too. They're very confused. They say it's cell death, but also grow better.
Keep only what sparks joy.
Yeah, exactly. So yes, the phenotype, keep in mind that this is overexpression. Like I really overexpress the protein. If I express less, it's not that cell death. It's slightly less and less and less. So it's like sometimes you just dump a lot of proteins somewhere to see what's happening, but it's not actually the biological real.
Fascinating.
Awesome. Thanks everyone for your questions.